Hello developmental cell, congratulations to your 10-year anniversary. And hello Angelica, many thanks for picking our paper. Now our paper is the work of 4 different authors. The first one is Mark Petronczki, who performed the key experiments for this study when he was a postdoc in my lab at the IMP here in Vienna. The second author is Michael Glotzer from the University of Chicago. The third one is Norbert Kraut from Boehringer Ingelheim, also here in Vienna. And the fourth one is myself. My name is Jan Michael Peters and I work here at the IMP in Vienna. The 4 of us share a deep interest in understanding the molecular mechanisms of cell division, and in this particular study we were interested in understanding the role of a protein kinase known as polo-like kinase or PLK1 in cytokinesis. Now it was clear from studies in budding yeast that polo-like kinase has an essential role in cell division, but it was very difficult to address if PLK1 also has such a role in mammalian cells, because in these cells PLK1 also has important functions at the beginning of mitosis, and inhibition of this function leads to an arrest of cells early in mitosis, and therefore cells would never progress to the stage of cytokinesis if PLK1 had been inactivated, and therefore these cells could never be studied in the absence of PLK function. Now the solution in our case to this particular technical problem was provided by Norbert Kraut and his colleagues at Boehringer Ingelheim, who had developed a very selective and potent inhibitor, a small molecule compound of PLK1, from their side with the main motivation to evaluate the suitability of PLK1 as a potential anti-cancer drug target. Now for us, this compound, BI-2536, provided the unique opportunity to allow cells to progress to the initiation of chromosome segregation, and then to treat these cells so that PLK1 activity would be very acutely inhibited at the onset of chromosome segregation. So if these were the separated sister chromatids, we now would be able to follow cells up to this stage in anaphase, where the mother cell normally begins to initiate cytokinesis by assembling a contractile ring and allowing furrow ingression. And now we could ask if at this stage or shortly before this, inhibition of PLK1 would interfere with either chromosome segregation or cytokinesis. Now in fact we discovered that inhibition of PLK1 at the onset of chromosome segregation had a very profound effect on the initiation of cytokinesis. And in an elegant series of experiments, Mark discovered also the mechanism behind this. Mark found that the recruitment of an enzyme to the so-called mid spindle, the enzyme is being called ACT2, depends on PLK1 activity, and this enzyme ACT2 had already been shown to be an activator of another enzyme called Rho A, a small GTPase, which sits here at the cortex of the dividing cell, where its activation leads to assembly of the contractile ring and thereby to the initiation of cytokinesis. So Mark could delineate a pathway by using BI-2536, which showed that PLK1 activity is needed to recruit ACT2 to the equatorial plane of the dividing mother cell, and that this signal would then activate Rho A and thereby specify the position of the cleavage furrow and the initiation of cytokinesis. Now in summary, I think this paper published in Developmental Cell is unusual for a number of reasons. One of them is that it shows, I think in a quite remarkable way, how highly conserved some of the essential molecular mechanisms can be from budding yeast to mammalian cells. In this case, despite the fact that cell division occurs quite differently in yeast cells, namely by budding, as the name implies, from how cell division occurs in mammalian cells. The second reason for why I think this is an unusual paper is because it illustrates really nicely the potential power of what is often called chemical genetics, where a small molecule inhibitor is used much in the way of how normally a temperature-sensitive allele or a mutant would be used in a genetic model organism. And then finally, and I think quite importantly, the study also is unusual because it illustrates how a collaboration between an academic research lab and the pharmaceutical industry can provide really unique opportunities that otherwise would simply not exist. for us, the availability of BI 2536 really allowed experiments that we could not have performed with any other system. And conversely, we like to believe that our work on the basic functions of PLK1 in cell division also might have contributed to the decision in the pharma industry to pursue PLK1 as a potential drug target in anti-cancer therapy. And I personally think if this would at one point in the future lead to a situation where this research could benefit cancer patients and would help to prolong their lives, then that obviously would really add a completely new dimension to our otherwise very basic work in cell biology. Thank you.